Afternoon, everyone. I'm Sue O'Shea. I'm a professor here at the University of Michigan in the Department of Cell and Developmental Biology, and I also have a cognate appointment in the Department of Psychiatry. So what I wanted to do this afternoon is talk to you a little bit about stem cell biology here at U of M, particularly about our core lab that's been established in the Department of Cell and Developmental Biology for over 15 years now. So stem cells, of course, come in many different flavors. There are pluripotent stem cells that are derived from early embryos and from skin samples that are called induced pluripotent stem cells. There are stem cells that are resident in many of the body organs that can be coaxed to differentiate into various differentiated cell types. Stem cell biology is a particularly important part of university research because it allows us to, first of all, study human differentiation and development in a way that we've not been ever able to do that before. It also allows us to think about making replacement cells for cells and tissues that have been lost due to injury or disease or aging. Some of the most important things that we're doing, I think, is using stem cells to model human development. For the first time, we can really look at how a neuron differentiates from the early embryo, or how a stem cell can form a cardiomyocyte or a bone cell or a muscle cell. So these are some of the things that are really efficiently differentiated from stem cells. One of the goals of stem cell biology early on was really to differentiate cells for replacement therapies, and that's frankly been less successful than using stem cells as models of disease or of development. So one of the projects that I really wanted to tell you about today has to do with studying bipolar disorder. And of course, bipolar disorder is a mood disorder that's characterized by really high highs, euphoria or irritability, and it's inevitably followed by lows or depressions. So obviously there hasn't been a way to study bipolar neurons in a tissue culture dish to really understand what goes wrong in bipolar disorder. So the ability now to take a small patch of skin from an individual with a diagnosis of bipolar and literally turn back the biological clock so these skin cells behave as though they were derived from stem cells, and they have the ability then to form all the cells and tissues in the human body. And that's a really important characteristic of stem cells. So what we've done with the bipolar neurons is to take them from skin samples of people who've been diagnosed with bipolar disorder, turn back the biological clock, put them into a tissue culture dish, and study their differences between the bipolar cells and the control cells. And we found a number of differences between them. And the goal, of course, is because bipolar is diagnosed in the late teens or the early 20s, obviously things have gone wrong before then. And the ability to take stem cells from their pluripotent or very primitive stage to an adult neuron is really very important to allow us to pinpoint when exactly during their differentiation things start to go wrong. And the hope, of course, is that we'll be able to use this information to really figure out new treatments and new medicines and new approaches to treat bipolar disorder. So the core lab is in the Department of Cell and Developmental Biology and has been since 2002. We received an NIH grant that funded us for about 5 years. The purpose of the stem cell core is really to help people transition their research from cell lines or perhaps animal research to using human stem cells. So toward that end, we've got great equipment and we've got staff that can help train and help people differentiate their cells into various cell types.